Sorry for being a little late today. Can somebody please confirm you can hear me?
Confirming.
Thank you. Let's see, where's our syllabus? All right, let me share my screen here. Go to where we are in the syllabus or the schedule. Today is February 5th, and we are supposed to be starting chapter 5 today. We're not quite there, I don't think. There is a lab today, lab 6. Any questions about what we're doing? Chapter 5, where were we? Oh, actually, we started chapter 5 last time. All right. There we go. So last time I had talked about the energy of activation and stated that whenever there's enough energy, the reactants will be changed into the products. We're talking about an enzymatic reaction. And when there isn't an enzyme around, you need a large amount of energy to get the activation energy without an enzyme. However, if there is an enzyme around, you need a less amount of energy, only that much there to get the reaction to proceed. And I told you that, like, for example, glucose breaking down into CO2 and water, you need the temperature of a blowtorch to get that to happen, which is something like 1,000 degrees. Whereas with an enzyme around, this can happen at 37 degrees Celsius or 98.6 degrees Fahrenheit inside your body for glucose to break down into CO2 and water. Any question about any of that? Okay, enzymes are biological catalysts. They speed up a biochemical reaction. They can speed up a biochemical reaction by 10 to the 8 to 10 to the 10th fold. So what is that? That's close to a billion times. Obviously, that's more than a billion, 10 billion, but there's 1/10 of a billion. Enzymes are encoded by genes. So we get these in our cells because they're encoded by our genes. We have enzymes specific for a single chemical reaction. This is a little bit of an oversimplification. As you know, we have some drugs which mimic binding to a enzyme similar to the substrate. But generally, enzymes are specific for one substrate. The specificity is due to the enzyme's three-dimensional shape, allowing an enzyme to act specifically by binding to and then changing one substrate into a product. And that's because the enzyme has a specific shape and it'll bind only to that one specific substrate. Now, like I said, that's a little bit of an oversimplification. As I stated, we do have some drugs that can mimic a substrate and then bind to the enzyme. But generally, it's pretty specific. One enzyme binds to one substrate, and then another substrate, you just need another enzyme to bind to it. Enzymes are not used up by the chemical reaction, and they are not altered in a chemical reaction, meaning that once an enzyme is done changing a substrate into its products, the enzyme is now free to bind to another substrate and then convert it into the product. Any question about any of that? How enzymes do this is they actually form an enzyme substrate complex. This orients the bonds in the substrate, increasing the probability of a chemical reaction, changing the substrate into the chemical bonds of the product. And this is what lowers the activation energy, the enzyme substrate complex forming, putting stress on the bonds of the substrate, increasing the likelihood that the bonds of the complex will form. Any question about any of that? All right. Enzymes normally end with ASE or ACE. So when we're talking about catalase, isomerase, ligase, we'll talk about lysase and catalase later in the term. These are enzymes because they end with ACE. You don't need to know these specifically, but you should know that most enzymes end with ACE. The only exception are enzymes that were named before this convention became established. Like renin is an enzyme that was discovered in the stomach, converting milk into liquid milk into, I don't know, solid milk or something like that, whatever you call solid milk whey. And that allowed babies, for example, to have the milk in their stomach longer, and then they would be better at digesting it. But renin is a enzyme, and it was named before this enzyme convention ACE was ever established. Any question about any of that? Enzymes do not work in a vacuum. Here we have an enzyme, you don't need to know the name of it, and it's getting together with either a coenzyme or a cofactor to make the complete functioning enzyme. Here we have an enzyme binding with a coenzyme. A coenzyme is a small biological molecule like NAD plus or FAD. We'll talk about these coenzymes in a little bit in today's lesson. And the enzyme gets together with the coenzyme to make the functional enzyme. And that's what allows the enzyme to be the correct shape to bind to one specific substrate. A cofactor is a non-protein portion that gets together with an enzyme to make the complete enzyme. And usually cofactors are simply just elements like iron, zinc, magnesium, and then they combine with the enzyme to make the finished functional enzyme. And the element, when it binds to the enzyme, it changes the three-dimensional shape of the enzyme, giving the enzyme this portion here, it's called the active site, which then binds to the substrate. Any question about any of that? Here are some important coenzymes, NAD plus, FAD. We'll talk about these two in aerobic respiration, this lesson. So you do need to know that NAD plus and FAD are coenzymes. You need to know them because they're coenzymes in aerobic respiration. We're going to talk about aerobic respiration a lot today. You don't need to know the rest of the coenzymes. NAD, P plus, which is a coenzyme in photosynthetic organisms. FMN, a flavin mononucleotide. I don't even know where this coenzyme works. It's similar to FAD. It's a riboflavin derivative. And then coenzyme A, I probably should make it required for you because it is used in aerobic respiration, but a little bit later than NAD and FAD, or is a little bit earlier. Let me think about that. It's actually a little bit earlier than most NAD and FAD. I suppose there is some NAD in aerobic respiration, which is used right away before coenzyme A. Anyways, you don't need to know coenzyme A. We will talk about it briefly in aerobic respiration, but you just need to know the first two. Any question about that? All right. As I mentioned, enzymes can work with a coenzyme to make the functional enzyme, which then has an active site. The active site binds to one and usually only one specific substrate. That's a little bit of an oversimplification, but for our purposes is good enough for this lesson. And that's why the enzyme can bind to the substrate, forming the enzyme substrate complex, allowing the enzyme to fit the substrate, sort of like a key fits one lock and then can unlock the lock. And that puts stress on the chemical bonds of the substrate, converting the substrate into the products. The product then falls out of the enzyme, and then this enzyme is then free to bind to another substrate. And this is called the lock and the key model of how an enzyme functions, where the enzyme has a specific shape, three-dimensional shape, having an active site that binds to one specific substrate, putting stress on the substrate, making the substrate, the chemical bonds of it change into the chemical bonds of the products. Any question about any of that? Here we're looking at the hand in the glove model of how an enzyme works. And there's three things you can detect from this model of how an enzyme works, where an enzyme here has a three-dimensional shape where the active site right here binds specifically to one substrate right there. And what can you notice? There's three things you can notice going on in this, I don't know, chemical reaction. Can anyone state one of the two, which is very easy? The third is more difficult to see, but there's three easy things, or two easy things, sorry. Nobody notices something? Well, what's the size of the enzyme compared to the size of the substrate?
One thing that I'm noticing from here is that it almost looks as if the enzyme is engulfing the substrate.
Do you mean it's changing its shape by engulfing?
Yeah, it seems
Yeah.
like it's kind of curling around it.
Yeah, that's one of the things that the hand in the glove model says, and that is the enzyme is a glove, which then changes its shape to fit the hand. And that's
Mhm.
why the enzyme can then bind specifically to the substrate. The lock and the key model is a little oversimplified, that the enzyme actually changes its shape to fit to the substrate. And then the other thing that's very easy to detect is that the enzyme is much larger than the substrate. The other one, which is difficult to detect, if you look here and you look there, can anyone notice a difference in the substrate? This substrate's a little more flat than that substrate. And this I like to call the shapeshifter's hand into the glove model. And that is, yes, the glove is changing its shape to fit the hand, but the shapeshifter's hand is actually changing its shape to fit the glove. And those of you who've had Star Trek, if you remember Odo was a shapeshifter, you know what the heck I'm talking about. The shapeshifter's hand is changing its shape to fit the glove. Any question about any of that? All right. Enzymes do not work in a vacuum. I've already stated that. They do need either some enzymes need a cofactor or a coenzyme. They also need to be at the correct temperature, the correct pH, the correct salinity. Generally, presence of heavy metals have to be absent. Heavy metals can bind with an enzyme and inactivate it. Alcohol generally can't be present. Also, alcohol can change the shape of the enzyme and then inactivate it. UV radiation can also inactivate the enzyme. And all of these temperature, pH, salinity, the presence of heavy metals, the presence of alcohol, the presence of UV radiation can cause the protein of an enzyme to denature, changing the enzyme to lose its three-dimensional shape, taking on another shape, and that can denature the protein, making the enzyme may not function. It still has a three-dimensional shape, but it's a different shape, and then the enzyme cannot happen. The substrate concentration can also affect the amount that the enzyme will work. Generally, when the substrate concentration is very low, the substrate isn't going to bind very often to the enzyme. And so if you increase the substrate concentration, you can increase the amount of enzyme activity up to a point, and we'll talk about why it's only up to a point in just a little bit. Enzymes can also be inactivated by poisons, so most enzymes don't want a poison around, like cyanide or fluoride can inactivate the enzymes. Cyanide or fluoride are two examples of poisons which inactivate enzymes. We'll talk about protein denaturation in just a little bit. You should also realize that enzymes can undergo inhibition, where inhibitor can inactivate the enzymes. There's two types of inhibition, competitive inhibition and non-competitive inhibition. And we'll talk about these in, I don't know, in 2 or 3 slides. And then lastly, we'll talk about feedback inhibition, where an enzyme can be influenced on its activity level by feedback inhibition, where when you get enough feedback, the enzyme will stop working. And then when the feedback is not there, the enzyme will be working at full force. Any question about any of that? All right. Enzymes can be inactivated, or their amount of enzyme activity can be decreased by being at the incorrect temperature and the pH. At an extreme temperature, either too cold or too hot, the enzyme will totally lose its normal three-dimensional functional shape. The protein will take on another shape, but still in 3 dimensions, but it no longer has the active site. Let's say right there is the active site, and I don't know where that would even be here, here, or here. And there's no longer an active site, and this enzyme cannot function at all. When you have slightly different temperature, let's talk about an enzyme in our body. It's going to work best at 37 degrees. If you slightly cool it or slightly rise it, the enzyme will begin to become denatured, will not be a perfect three-dimensional shape, but it will be having some similarity, and the enzyme will have some activity. And then as you change the temperature more and more, the enzyme will lose more and more activity until the enzyme eventually gains zero activity, which would be, I don't know, either like refrigeration temperature or 0 degrees temperature for an enzyme in us, and then usually something like 65 degrees Celsius, certainly boiling temperature, which would be 100 degrees Celsius, the enzyme will become totally denatured. Most of our enzymes will become denatured much lower temperature than boiling, something like 65 degrees Celsius. And I don't know what that would be in Fahrenheit. We can go to a web page here. Hopefully it understood me. Around 149 degrees Fahrenheit. Not terribly meaningful. So when we look at the temperature, an enzyme in us will be optimal at 37 degrees Celsius because that's our body temperature. And then as you go a little cooler or a little warmer, the enzyme will begin to be denatured and will have less activity. So 30 degrees, it'll be less active than, it looks like about 42 degrees. It'll be less active than 37. And then you go cooler, 25, it'll be less active. 20 degrees, even less active, 15 degrees, even less active, and then down to refrigeration temperature about 10 degrees Celsius or cooler, our enzymes will have no activity. And here, this enzyme is going to be no activity around 50 degrees Celsius. Any question about any of that? So an enzyme has an optimal temperature, and for our enzymes inside our body, their optimum temperature will be 37 degrees Celsius. That isn't true for the enzymes in our skin. Their optimal temperature will be closer to around 30 degrees. And then the graph would be more like here. This is true for pH too, where an enzyme has an optimal pH. Here, this enzyme, its optimal pH is around pH 5. And as you start decreasing the pH, you get less and less activity by the enzyme until you get zero. There's no enzyme activity. Or here, you increase the pH, and as you increase the pH, you get less and less activity until you reach about pH 10, and then you have no enzyme activity. Remember, if you have no enzyme activity, then the enzyme is totally denatured. So where would you find an enzyme in humans that has an optimal pH of around pH 5? Anyone have a guess? Those of you who have been in what is that called? The biology class, similar to microbiology, it's not 160, it's some of the 200 level. I forgot the name of the class. Gosh, whatever it is. PA? You know what I'm talking about? RA? Anatomy and Physiology. Sorry, AP. That's what I'm trying to say. If you've had anatomy and physiology, you should know this. What is an enzyme that has an active optimal pH working around pH 5? Where would this enzyme be in the human? It's not the stomach because the enzyme would have an optimal pH between 1.5 and 2 if it was in the stomach. And that would be something like renin. That's the enzyme
Well,
that
the
smell.
pH of blood is like about 7. So,
Yeah,
and
pH of blood is a little above 7. It's like 7.2, 7.4. So it's not the blood, you're right.
is it the intestines? Or is that
No,
acidic because of the bile?
yeah,
I'm
the
in A and
intestines
P.
actually become slightly uh, acidic, slightly basic in the intestines. No, it's the pH of the skin. It's around pH 5, actually can get as low as pH 4.5. So this enzyme here, if it's from a human, I'm going to guess it's an enzyme in the skin. Another thing that can influence the amount of enzyme activity, and I talked about that, is the substrate concentration. If you have the substrate concentration very low, the amount of enzyme activity will be very low because the chance of the enzyme coming together with the substrate will be low because the substrate level concentration is very low. And as you increase the amount of substrate, the amount of enzyme activity will increase because the chance of the enzyme meeting the substrate will increase as you're increasing the substrate concentration. That will happen up to a point until at this point here, when we increase the substrate concentration, the enzyme activity does not increase at all. And this happens because once you have all of the enzyme already bound to substrate, if you increase the substrate concentration further, it doesn't increase the enzyme activity any further because all the enzyme is already bound to substrate. So you can't increase the activity of the enzyme any further by increasing the substrate concentration. Any question about any of that? All right, let's talk a little bit about inhibition. There are, remember, two types of inhibition: competitive inhibition and non-competitive or allosteric inhibition. We'll talk about that one in just a little bit. Competitive inhibition is where the inhibitor competes for the active site with the substrate. So there's our enzyme, lock and key model. There's the active site. It binds specifically to one substrate. And the competitive inhibition inhibitor can bind to the active site instead of the substrate, competing with the substrate for the enzyme. We do have some drugs which act as competitive inhibitors, inactivating the enzymes. So a competitive inhibitor is one that binds to the active site, competing with the substrate for the active site. On the other hand, a non-competitive inhibitor, also called an allosteric inhibitor, is an inhibitor that binds to an enzyme at a site other than the active site. And it's actually called the allosteric site. You don't need to know that name, but the non-competitive inhibitor binds to the enzyme at the allosteric site, and that changes the three-dimensional shape of the enzyme so that the active site no longer binds to the substrate. And that's how a non-competitive inhibitor inhibits an enzyme. Any question about any of that? Let's talk a little bit about feedback inhibition. Feedback inhibition is often found in cells where an inhibitor can be inactivated, and usually the inhibitor is inactivated by the end product of the reaction. So here we have a chemical reaction where a substrate is converted by enzyme 1 into intermediate A, which is converted by enzyme 2 into intermediate B, which is converted by enzyme 3 into the end product. If we have no end product in the cell, there's nothing binding to the first enzyme, and then enzyme 1 makes intermediate A, which then enzyme 2 makes intermediate B, which then enzyme 3 makes into the end product. Once the end product concentration in the cell starts increasing, the end product will begin binding to enzyme 1 as a non-competitive inhibitor, altering the shape of the enzyme so that the enzyme will not bind to the first substrate. As more and more end product builds up in the cell, more and more of the enzyme will be bound to the end product, and more of enzyme 1 will become inactivated until you have a bunch of end product in the cell and then all of enzyme 1 is inactivated. Once all of enzyme 1 is inactivated, intermediate A will no longer be made, so intermediate B will no longer be made, and then the end product will no longer be made. Any question about any of that? As the end product is no longer being made, it'll be slowly used up by the cell. Usually the end product will do something else in the cell besides just binding to enzyme 1. Like it usually has a biochemical function in the cell, and it might be metabolized or something like that. My point is that the end product will decrease in concentration in the cell, and then more and more enzyme 1 will become active as the end product falls off of enzyme 1, and then we'll begin making intermediate A, making intermediate B, and then resuming making the end product once again. And that is feedback inhibition, which is often found in cells. Why do you think in a cell we have the end product tending to be the molecule which inhibits the first enzyme? Why doesn't the end product inhibit, I don't know, the third enzyme or the second enzyme? Why is it the first enzyme? And then why is it the end product also the molecule that acts as the allosteric inhibitor? Why can't it be intermediate A or intermediate B? Any ideas? Anyone have a guess? Nobody's going to take a guess. I'll give you an extra credit point if you get it right. If you partially get it right, I'll give you an extra credit point.
Isn't it because they have different shapes per each enzyme? Like they have their own unique one?
That is true, but that's not why it's the end product that's binding to the first enzyme. Like your end product could bind to the second enzyme or the third enzyme. And it doesn't really explain either why intermediate B isn't the molecule shutting down enzyme 1. It's the end product. It's because if the end product shuts down the first enzyme, you don't make intermediate A, you don't make intermediate B. And
It's
that's
a negative feedback cycle.
Yeah, it's a negative feedback cycle. It's just that this is the most energy efficient feedback cycle. If you had intermediate A shutting down enzyme 1, you still have to make intermediate A, so it's not as energy efficient. And then if you had the end product binding to enzyme 2, once again, you still have enzyme 1 making intermediate A, and it's not as energy efficient. And the cell wants energy efficiency, and that's why the cell tends to use the end product, which tends to bind to enzyme 1, shutting it down. And that's why we see feedback inhibition usually being this cycle, where the end product shuts down enzyme 1, because it's the most energy efficient feedback inhibition the cell can have. All right, let's talk a little bit more about enzyme. As you notice, I occasionally talked about enzymes as if they are a protein. I mentioned, what was it back here? Was it here? Someplace here. That the enzyme can be denatured as the protein begins losing its form, and when the protein totally loses its form, the protein is totally denatured. However, most enzymes are proteins, but not all enzymes are proteins. Some enzymes are actually made of RNA, not protein. And these enzymes have a specific name, ribozymes, where the RNA makes an enzyme. The RNA actually cuts and splices RNA or another molecule, similar to the way a protein enzyme can cut and splice a molecule. We'll talk about splicing in just a little bit. Splicing is where you cut the RNA at certain specific points, and then usually you splice the RNA pieces back together. So these ribozymes function as enzyme in that they have active sites where the ribozyme has an active site that actually binds to the substrate. And then the ribozyme is not used up or modified in a chemical reaction. Once the RNA enzyme converts the substrate into the products, the RNA enzyme is then freed from the complex, and then it can bind to another substrate, converting that substrate into products. Any question about any of that?
So they are an enzyme, they're just a non-protein enzyme.
Yes, that's why they're called the ribozyme. They're an RNA enzyme, but they are an enzyme. Right there, it says it. It's just an RNA enzyme, not a protein enzyme. Most enzymes are protein, but there are a few which are RNA. There's only one which we're going to mention in this class. Tutorial does not mention it, but when the ribosome links the amino acid together in a growing polypeptide to eventually make the protein, that happens by the ribosome removing the amino acid and then attaching it to the growing polypeptide chain. That is actually happening by an RNA enzyme. It's the RNA portion of the ribosome which does that. Like I said, tutorial doesn't mention it, but this is the only RNA enzyme we will discuss in this class. And I guess I do discuss another one when we're talking about eukaryotes, when they splice the RNA. That tends to happen by an RNA enzyme. You don't really need to know that, but this one we will talk about in this lesson, where the ribosome can remove the amino acid that was just added to the ribosome and then take that amino acid and add it to the growing polypeptide chain. That happens because of a ribosome. It's the RNA portion of the ribosome that does that. And that's one RNA enzyme we will discuss, at least in today's lesson. All right, any question about ribozymes? Sorry, a ribosome is the protein manufacturing site on a cell. A ribozyme is a non-protein enzyme. All right, let's talk a little bit about metabolic diversity. Organisms have metabolic diversity, and we can split all organisms, including microbes, into the following requirements: where they get an energy source and where they get their carbon source. When we divide microbes, as well as actually any organism, into where they get their energy source, we can divide or classify organisms into phototrophs and chemotrophs. Phototrophs is a cell or a microbe which derives its energy from light. Photo, light, troph, energy source. And then chemotroph, these organisms or cells derive their energy needs from a chemical compound, whether a chemotroph. And this chemical compound can be an organic compound, like glucose, and we're that, or it can be an inorganic compound, like iron or nitrate, nitrite, sulfate, hydrogen sulfide gas. These inorganic compounds for deriving the energy source usually happen, if not entirely happen, by a chemosynthetic bacteria. And we will talk a little bit about that. They are bacteria that derive their energy from an inorganic chemical compound. Any question about any of that? And then we can derive an organism's carbon source depending on where the organism obtains its carbon. Autotrophs are organisms that get their principal carbon source from CO2, either dissolved in the air or in the water. A heterotroph is one which gets its carbon source from an organic chemical molecule, such as glucose. And we are a heterotroph because we get our carbon source from an organic chemical compound, such as glucose. As you know, green plants are an autotroph because they get their carbon from CO2 in the air. But we also have bacteria and archaea, which are autotrophs, and they get their principal carbon source from CO2. I should mention plants are a phototroph, and then we have photosynthetic bacteria, which can also get their energy needs from light. And then we have chemotrophs, bacteria like E. coli, which gets its energy from an organic chemical compound such as glucose. Any question about any of these? All right, when we're talking about metabolic diversity, if you get confused, just divide it into where it gets its energy source from and where it gets its carbon source from. And that will simplify the, the metabolic diversity, what the troph is. And then once you have that, you can then combine them together, putting the carbon source and the energy source together, and then classify the organism on where they get their energy source as well as where they get their carbon source from. And then we get 4 different trophs: a photoautotroph, a photoheterotroph, a chemoautotroph, and a chemoheterotroph. The photoautotrophs, I think, are the easiest to explain. A photoautotroph gets its energy need for light. That's why it's a phototroph, energy source from light. And it gets its carbon needs from CO2. That's why it's an autotroph. And everybody knows green plants are photoautotrophs. We also have green photosynthetic bacteria, like the cyanobacteria, which are photoautotrophs. Any question about any of that? The next easiest one to explain are the chemoheterotrophs. And that's easy to explain because we are a chemoheterotroph. We get our chemical needs from glucose or some organic chemical compound. And that means we get our energy source from a chemical compound, such as glucose, chemotroph. And then we get our carbon source from an organic compound such as glucose, and that's why we are a heterotroph, and that's why we are a chemoheterotroph. Oftentimes, chemoheterotrophs get both their energy source and their carbon source from the same chemical compound. For example, humans get both their energy needs and their carbon from glucose, assuming you have a diet high in sugar or glucose. Bacteria, much of the bacteria are this way, like E. coli. Same thing. The fermentative bacteria, like the one making yogurt, same thing. Get their energy source from a chemical in the yogurt or the milk, probably the milk sugar. And then the organic compound glucose or the lactose in the milk gives its carbon source. Animals, protozoa, fungi, many bacteria are chemoheterotrophs. Any question about that? The next easiest one to explain are the chemoautotrophs. These are chemotrophs. They get their energy source from a chemical compound, but this chemical compound is an inorganic chemical compound, such as sulfate, hydrogen sulfide gas, nitrate, or iron. These organisms tend to be the chemosynthetic bacteria or archaea, deriving their energy needs from an inorganic chemical compound. There are no chemosynthetic animals, plants, or fungi. So you only need to know about them in archaea and bacteria. And they get their carbon source from CO2, at least the chemoautotrophs. That's why they're an autotroph. They get CO2 as their main carbon source. The iron oxidizing bacteria, as well as nitrate oxidizing bacteria, is an example of a chemoautotroph. And actually, you guys have all seen this iron oxidizing bacteria performing chemoautotroph when you lift up the water tank of the toilet and you see that brown or yellowish sludge on the back of the water tank. That's an iron oxidizing bacteria getting their energy needs from iron in the water, and CO2 was dissolved in the water. It'll obviously come from the air, but it's dissolved in the water. So you've all seen an iron oxidizing bacteria. You just didn't know it at the time, but I'm telling you now. And that is a chemosynthetic bacteria. Now, the difficulty is some chemosynthetic bacteria are chemoautotrophs, and some chemosynthetic bacteria are chemoheterotrophs. It depends on where the organism gets its carbon. If the chemosynthetic bacteria gets its carbon source from CO2, this is a chemoautotroph chemosynthetic bacteria. But if the chemosynthetic bacteria gets its carbon from an organic compound like glucose, then this is a chemoheterotroph chemosynthetic bacteria. And so the chemosynthetic bacteria are split into two groups, the chemoautotrophs and the chemoheterotrophs. Any question about any of that? All right, the most difficult one to explain are the photoheterotrophs. They get their energy source from light, why they are a phototroph, and they get their carbon source from an organic chemical compound such as glucose, why they are a heterotroph. The green and the purple non-sulfur bacteria are photoheterotrophs. How many of you have actually even heard of the green and purple non-sulfur bacteria? Anyone? Probably not. Nobody's saying anything, so I'm going to assume nobody has. And like I said, these organisms you're not terribly familiar with, and that's why the photoheterotrophs are the most difficult to understand and describe. All right. Any question about the four trophs? Photoautotrophs, photoheterotrophs, chemoautotrophs, and the chemoheterotrophs. In the nutritional classification of organisms, you need to know everything in the red box. So we can divide all organisms into their energy source, and if they get their energy needs from a chemical compound, we call them a chemotroph. If, on the other hand, they get their energy needs from light, we call them a phototroph. You can then split the phototrophs into where they get their carbon source. If they get their carbon source from CO2, they are a photoautotroph. If, on the other hand, they get their carbon source from an organic chemical compound, such as glucose, they are a photoheterotroph. And likewise, you can split the chemoheterotrophs, or the chemotrophs into where they get their carbon source. If they get their carbon source from CO2, either dissolved in the air or the water, they are a chemoautotroph. If the organism gets their carbon source from an organic chemical compound, such as glucose, they are a chemoheterotroph. You don't need to know this, but you can split the chemoheterotrophs into where the final electron acceptor is. If it's oxygen, most animals, fungi, protozoa, most bacteria, then they can engage in aerobic respiration. And oxygen is the final electron acceptor. But there are other organisms where oxygen is not the final electron acceptor. And you can split these. These will be the non-aerobic respiration organisms. You can then split these organisms into whether they use an organic chemical compound for the final electron acceptor, or they use an inorganic compound for the electron acceptor. You don't need to know if fermentative bacteria have an organic compound as the final electron acceptor, but in anaerobic respiration, where the electron transport chain will accept the electron and then pass it to an inorganic chemical compound. And this would be anaerobic respiration. Don't need to discuss that any further. Don't need to discuss that one any further. We could talk about the photoautotrophs. You can split them into two groups, whether they are oxygenic, releasing oxygen in photosynthesis, or whether they are non-oxygenic and they do not release oxygen in photosynthesis. Like I said, anything beyond the red you don't need to know. Come on. All right, let's talk a little bit about energy production. The nutrient molecules have energy associated with the electrons that form their chemical bonds. And it's from these chemical bonds, when we break them, that we can obtain the energy from nutrient molecules. In cells, energy is obtained, at least in our cells, from catabolic reactions. And that energy is stored in the bonds of our nutrient food molecules, like glucose. ATP is the energy carrier of cells. Usually when a cell needs energy, it will take that energy from the energy stored in ATP. ATP has high energy bonds, actually has two of them, where the phosphate is bound to ATP in a high energy bond. That high energy bond is unstable, and when it is broken, it releases a lot of energy. Some of that energy can be used for the cell's energy needs. So ATP is called the energy carrier of cells. How energy is produced in our cells is by oxidation reactions, oxidation reduction reactions. Oxidation is the loss of electrons. Reduction is the gain of electrons. Oxidation and reduction reactions are always coupled, meaning you have one molecule or atom or ion which loses an electron, and another molecule, atom, or ion which gains the electron. So one molecule loses the electron and it will be oxidized. The other molecule gains the electron and it will be reduced. An exact example of that is copper +1 reacting with iron 3+ can be converted into copper 2+, meaning this copper loses an electron, so gains a positive charge. The iron 3+ gains an electron and then becomes reduced, meaning instead of +3, it's now +2. And so iron is reduced, copper is oxidized. Any question about any of that? An easy way to remember oxidation reduction reaction is to say the mnemonic device, Leo the lion goes grr. Leo, lose electron oxidation. So Leo the lion goes grr, grr, gain electron reduction. Any question about any of that? In our cells, oxidation reduction reactions are occurring in a biological system, and usually in biological systems, a proton, an H+ ion, is usually removed when the electrons are removed. And then the proton, the H+ ion, tends to follow the electron. Remember a hydrogen atom, all it's going to be is a hydrogen plus and one electron -1. That plus is a +1. And so a proton, a H+ ion, is usually removed with an electron. Here we have a molecule that's actually losing 2 electrons, so it will become oxidized. NAD plus will gain these 2 electrons, shown here, which is then NADH. Instead of NAD +1, it's NADH. And so NADH is going to be reduced from NAD +1. And this molecule, besides losing 2 electrons, loses 2 hydrogen ions. One of them actually binds to the NAD+ to become NADH. The other H+ is just following the NADH. So in reduction and oxidations, we usually have the hydrogen following the electron, and then the reduction, the gaining of the electron, that molecule tends to gain an H+. Any question about any of that? And these H+ tends to happen in biological systems. Biological redox reactions are used in catabolism to extract energy from nutrient molecules. And let me say that redox, I don't think I did, redox is a simplification for oxidation reduction. Redox for reduction, and then ox for oxidation. So they sort of reversed it, but redox means oxidation reduction. We have highly reduced compounds. That's a molecule with many hydrogen ions. They are degraded, catabolized into highly oxidized compounds. And glucose is a molecule with a lot of hydrogen. So it's a highly reduced compound, and it'll become oxidized into CO2 and water, which is a highly oxidized compound. Along the way, the energy released from glucose can be trapped and stored in ATP. So when you break down glucose, you can make ATP. And then these highly reduced compounds like glucose, they store a lot of energy in the chemical bonds of this molecule. And when we break these chemical bonds, we release that energy. And some of that energy can be used to make ATP. And then you store a lot of that energy in the bonds of ATP. Any question about any of that? All right, let's begin talking a little bit about aerobic respiration. Aerobic respiration is how our cells and many cells obtain their energy needs, where you take glucose, C6H12O6, combine it with molecular oxygen, exactly 6 molecular oxygens, and then you take 38 ADP molecules and 38 inorganic phosphate molecules to make ATP, and that results in a chemical reaction where you generate 6 CO2, 6 carbon dioxide, 6 water molecules, and 38 ATP molecules. These 38 ATP molecules store energy, and if the cell needs energy, it usually obtains it from ATP. Any question about any of that? This is a summary of the steps of aerobic respiration. So in one step, we're showing the summary where you take glucose, C6H12O6, 6 CO2 molecules, those 38 ADP molecules, and 38 phosphate molecules to generate 6 CO2, 6 water, and 38 ATP molecules. Any question about any of that? All right, let me just state briefly here, we'll talk about it later, that none of the CO2 generated by aerobic respiration comes from oxygen. All of that CO2 comes from glucose, and then all of this water, none of that comes from glucose. All of this water comes from the oxygen. And so in reality, we should have 12 water molecules being generated here, and you need to add 6 water molecules in here because we have 12 oxygen, and all of that oxygen atoms will end up as water. None of them end up as CO2. Any question about that? And that's why you need 12 water here, and then you had to add six oxygen there. All right, any questions about the summary of aerobic respiration? Aerobic respiration is frequently called cellular respiration. I don't really like that term, but it's one I'm forced to use because, well, biologists and the medical people use aerobic respiration synonymously with cellular respiration. However, let me point out that cells can undergo respiration, and that respiration can be aerobic respiration or it can be anaerobic respiration. So just remember that cells can perform anaerobic respiration, but when you see the term cellular respiration, we don't mean anaerobic respiration. We always mean aerobic respiration. And I know that is confusing. Like I said, I don't like the term cellular respiration, but I'm stuck using it because that's the way it's defined. When cells are making energy, they tend to make that energy and then store it as ATP. And they're obviously not making energy, they're getting energy from some source, like either the sun or a chemical. And some of that energy is used by redox reactions to make ATP. ATP is generated by phosphorylation. That's the phosphorylation is just the addition of a phosphate group to a molecule. That's what phosphorylation means, phosphorylating a molecule. And here we're phosphorylating ADP to make ATP, adenosine triphosphate. And let me blow this up a little bit here so you can see it better. This third phosphate is a high energy bond. Why it's a squiggly line. But second phosphate bound in ATP is also a high energy bond. And that's why it has a squiggly line there. If a cell needs energy, it will frequently take that energy by breaking down ATP and break this bond here that will release this energy for the cell's energy needs. If the cell needs further energy, it can then break this bond and then use the energy from that bond for its energy needs. Usually the cell only breaks this one, but sometimes it does break the second one. All right, so in the generation of ATP, cells have three mechanisms for making ATP. The first is substrate level phosphorylation, where phosphate from a molecule is just moved from that molecule onto ADP to make, this will be 3C, no phosphate, and then the phosphate moves to ADP to make ATP. So that is substrate level phosphorylation, where the phosphate moves from a molecule directly to ATP. This actually happens in fermentation and in glycolysis, the first step of aerobic respiration, where we use substrate level phosphorylation to make ATP. The second way cells have for making ATP is by respiration. And you'll note that I'm not saying here aerobic respiration or anaerobic respiration. I'm just saying respiration. And that's because it can be both aerobic respiration and anaerobic respiration. With respiration, we have the electrons moving down an electron transport chain. These molecules here are the electron transport chain, where the electron actually comes from a high energy molecule like glucose, and then it's brought to NADH or FADH2, which then deliver the electron to the electron transport chain. The electrons move from one molecule in the chain to the next molecule, and then from this molecule to the next, all the way down the chain. With aerobic respiration, the molecule will end up at oxygen. Along the way, the electron loses energy, so that the energy is lost in controlled ways. And that's how we can burn glucose in our cells and maintain our cell at 37 degrees Celsius. If you do not know, if you were to burn glucose, a cup of glucose right here, I don't know if you can see this, a cup, there would be so much energy released if I were to take a blowtorch to the glucose and then catch it on fire. There would be so much energy, I'd have to stand back at least 6 feet, maybe even further, because of all the energy being released at one point. In our cells, we release the energy in steps so that our cells will be a much lower temperature than if we were to release the energy at one time. And that's why our cells can do it at 37 degrees Celsius, which is 98.6 degrees Fahrenheit. Anyways, along the way, energy will be released, and in some of these steps, we'll talk about that later, ATP can be generated, and then respiration makes ATP. Any question about any of this? We'll look at this step in greater detail when we talk about aerobic respiration, which will be in our next lesson where we talk about it. We won't be able to get to it today. The third way that cells can make energy is by generating ATP in photophosphorylation. Photophosphorylation only happens in photosynthetic cells. So green plants can engage in photophosphorylation, where light actually releases electrons from chlorophyll, assuming the photosynthetic plant has chlorophyll. Most plants do. Actually, all plants do. But photosynthetic bacteria might have chlorophyll or could have something else. And the photons from the light release the electrons from chlorophyll or another photosynthetic pigment. And then that light energy will be converted into chemical energy like glucose via an electron transport chain. ATP and NADPH are generated. NADPH, similar to NADH, it's just a molecule we find in photosynthetic organisms, NADPH. However, the ATP and the NADPH are often used up to make sugar. And so photophosphorylation may not make ATP as the end product, it may make sugar as the end product. And we'll talk about that in our last 2 or 3 slides in this lesson, where photosynthesis makes either ATP or sugar. If it makes sugar, it doesn't make ATP. And then likewise, if it makes ATP, it doesn't make sugar. So those are the 3 ways that cells can make ATP: by photophosphorylation, shown by the organisms here. That's why the boys are so muddy. They're engaging in photophosphorylation. Actually, that's just a joke. The plants in the background, which are green, are engaging in photosynthesis. The boys do not engage in photosynthesis. The second way, by respiration, and then the third way, by substrate level phosphorylation. And I realize I have that backwards from the 3 ways here. I got the first way, substrate level phosphorylation; the second way, respiration; and the third way, photophosphorylation, which you only find in photosynthetic organisms. Any question about any of that? All right, if there's no questions, I think I'm going to end it here. And I will see you at 6:30 for the lab. Any questions? All right, bye.